am going to go ahead and get started with the public portion of my defense. And because I'm going to give my acknowledgements at the end, but some people will need to leave early, I want to take a minute now to thank everyone for coming and for all the ways that you have supported me throughout my process in graduate school. I'm going to give a land acknowledgement. We acknowledge that the state of Kansas is historically home to many Native nations and is the current home to 4 federally recognized Native nations: the Prairie Band Potawatomi, the Kickapoo Tribe of Kansas, the Iowa Tribe of Kansas and Nebraska, and the Sac and Fox Nation of Missouri in Kansas, Nebraska. K-State's status as a land-grant institution is a story that exists within ongoing settler colonialism and rests on the dispossession of Indigenous peoples and nations from their lands. These truths are often invisible to many. The recognition that K-State's history begins and continues through Indigenous contexts is essential. I'm not actually at K-State right now because I moved last year, so I'm giving my defense presentation in Minnesota Makose on the ancestral homeland of the Dakota people who were forcibly exiled from this land because of aggressive and persistent settler colonialism. And it's important to also acknowledge that in this historical moment, it is extremely apparent the ways that colonial projects and colonial violence continue to be ongoing here and across the world. Considering the topics of my defense today, I also want to acknowledge that colonialism and imperialism are major drivers of climate change, environmental contamination, and habitat destruction, and Indigenous peoples are disproportionately impacted by the consequences of these processes, while simultaneously safeguarding 80% of the world's remaining biodiversity. And given my discussion of sex and gender diversity, I also want to acknowledge that settler colonial violence against Indigenous peoples has been both justified by and escalated towards Indigenous cultural conceptualizations of gender beyond the man-woman binary, and that this violence continues to disproportionately impact Indigenous, queer, transgender, two-spirit, and gender diverse individuals. So this is an overview of my presentation, and I do want to note that if you've been to other biology dissertation defenses, mine is going to be a little bit different. In addition to my first 2 chapters, which focus on plant biology research, I am also going to talk about my 4th chapter, which considers how to create more LGBTQI+ inclusive classrooms and curricula. And although these topics are different, they are united by shared key concepts related to evolutionary biology that I will highlight throughout the presentation. So, given the topic of this presentation is on evolutionary biology, there are important key concepts that run through studies of evolutionary biology and also throughout my chapters related to the ways in which evolutionary biology is the study of diversification and adaptation of biological life on Earth over time and space. And the first of these key concepts that I'll introduce is variation. And variation is both a prerequisite for evolutionary processes, as they cannot occur in populations that do not have variation. And additionally, in response to evolutionary processes, species across the world have become immensely diversified in order to fulfill different niches within ecosystems and to adapt to the conditions of different ecosystems. Another key concept is response to context. When we think about evolutionary fitness, the fitness of any trait is always relative to the environment in which that organism exists. So the traits that make penguins well adapted to living in cold climates would be detrimental and not as adaptive if they lived in the neotropics where toucans are. The last key principle that I will cover for this introduction is the anthropogenic impacts on evolution. So since the Industrial Revolution, the amount of change that human activities have made to the biosphere has been so great that some people consider us to have entered a new ecological epoch, which is called the Anthropocene. I don't know who's unmuted, but if you could mute yourself, that would be awesome. And with considering the anthropogenic impacts, this has created new evolutionary constraints, challenges, and opportunities for organisms on Earth. And one example of this is rapid evolution that has occurred in the white-footed mouse, where populations that live in urban New York City have undergone genetic and physiological changes in the period of time since urbanization when compared to non-urban populations of the same species. And the information that I've learned about these 3 key concepts within evolution, as well as other key concepts, have led me to view both evolutionary biology itself and the study of it as something called a nature culture. And this is a concept that was coined by Donna Haraway and later further applied to evolutionary biology by Banu Subramaniam. And basically, a nature culture refers to the way in which science, nature, and human culture co-create each other. There are many examples of this, and I'm going to be discussing 2 of them. So when we think about the evolution of Poaceae, which is the grass family, this is an incredibly successful plant family, both in terms of the number of species that exist and the amount of terrestrial land that members of this species cover. And this success is due in part to the ability of Poaceae to both adapt to and modify ecosystems it exists in order to meet its needs. The success of Poaceae has also occurred in the context of coevolution with humans, and it has proved amenable to domestication, and many important food sources for humans are crops from the Poaceae family, and human movement has also enabled new introductions and invasions of the species. At the same time, Poaceae faces threats from climate change and habitat degradation, including Poaceae-dominated ecosystems such as grasslands and prairies. Another example of a nature culture is the scientific process itself, and I think many of us who are scientists are aware of how events that occur at the intersection of ecology and culture often cause our research to go in unexpected directions. In 2018, there was a severe drought in Kansas, and as a result, 2 different sets of graduate students in my program pivoted from their expected research to look at the effects of this unexpected drought on organisms on Kanza Prairie. This drought is, of course, occurring within the context of increasingly frequent drought due to anthropogenic climate change. Another example is the COVID-19 pandemic that had incredibly pervasive and destructive impacts as a result of the interaction of virus ecology and human behavior. And many scientists have to pivot away from their expected research in 2020, including Caleb Kruger, who gave a talk at a conference I recently attended, where he discussed that being unable to do fieldwork in 2020, he instead went through 50 years of existing data on sex ratios and sex determining mechanisms in turtles and ended up creating a database on this topic, which he also published. Additionally, experiences of systemic transphobia that impact many spaces and institutions in which we work motivated my collaborator, Kelsey Lewis, and I to put together a symposium discussing the topic of sex science and society as it relates to biologists and how we need to recognize our obligations in being responsive to these impacts. And this led us to a symposium last year, as well as the publication that I've screenshotted here. So as a reminder, these are some of the overarching concepts and perspectives that will be present throughout my work. And now I'm going to move into my first data chapter, which is titled Variations in Drought Tolerance Across Climatically Diverse Populations of the Wild Foxtail Millet Soteria viridis. So local adaptation is the process through which populations within a species experience different natural selection pressures depending on their local environments and differentiate from each other through adaptation to their local environments. This is especially important to consider when we think of plants, because plants are abiotic, or not abiotic, plants are sessile, and they cannot relocate physically. They cannot move to a new environment when conditions get stressful, and therefore they must rely on morphological and physiological mechanisms of response. The study organism in which I looked at local adaptation stress response is Soteria viridis, the wild foxtail millet. This is a wild relative of the domesticated foxtail millet, Soteria italica, an agriculturally important species in China and India. Soteria viridis has a wide native range across diverse climates. It has low intrapopulation variation and high interpopulation variation, and it also has a short annual life cycle and high rates of selfing. Additionally, it's a great plant species for genomic work because of the genome being relatively small and diploid, and it also has an available sequenced genome. In my first experiment, I asked whether climatically diverse populations of Soteria viridis varied in their drought tolerance. I looked at 9 populations from 8 different locations that received a variety of different amounts of precipitation on average per year, from 170 to 1260 millimeters. I germinated seeds from these populations under well-watered conditions, and when the plants were a month old, I began a drought treatment in which I kept them in the greenhouse and withheld water to allow the plants to dry down. During this process, I measured pot weight, fluorescence, and photosynthesis, and I also assessed correlations between population drought tolerance and 19 bioclimatic variables related to their location of origin. So, looking at the results from this experiment, to orient you to this figure, the y-axis shows how quickly plants stop photosynthesizing and result to drought stress, and the length of the pink bars show the number of days on average that each population took to stop photosynthesizing. So I did in fact find significantly different amounts of drought tolerance between populations, with the population from Afghanistan being the least drought tolerant and the populations from China, Portugal, and one of the Michigan populations being the most drought tolerant. When I looked at the relationship with these biochem variables, this is a lot of information on the slide, but the most important part is the p-values, and you can see that none of them are significant. So none of these 19 bioclimatic variables significantly correlated with the differences in drought response I observed. So that is not an explanatory factor for this outcome in this experiment. Something that might be partially explanatory would be the difference in root surface area to above ground biomass ratio. So existing research has shown that plants that have higher root surface area may be more able to withstand drought stress. And in my experiment, I used the ratio of root surface area to above ground biomass ratio due to differences in overall plant size between populations. So that's what's shown on the y-axis. And I did find significant differences by population, with one of the two drought tolerant populations I assessed having greater root surface area to volume ratio to above ground biomass ratio than the other two populations that were more drought sensitive. However, I didn't see this for the other drought tolerant population. So if this is explanatory of the differences I saw in drought response, it's not fully explanatory. The other question I asked in this chapter was, does plant age influence drought tolerance in Soteria viridis, and is this consistent across different populations? So to do this experiment, I picked two populations, one of which I'd found to be more drought tolerant and one of which I'd found to be more drought sensitive. I germinated seeds again under well-watered conditions, and this time I started the dry down experiment when plants were 3, 4, or 5 weeks old to look at differences in age. And again, I measured pot weight, fluorescence, and photosynthesis during drought treatment. So in these experiments, the figure on the left shows my results and the comparison between different populations at each age group to see if there was a population difference, and there was, and this confirmed what I'd seen in my previous experiment with more drought tolerance in the population from China than the one from Russia. And this panel on the right shows a comparison within population across different ages, and I did find significant differences, with the plants that were the oldest at the start of the experiment being the least drought tolerant compared to the other populations. Something else that I assessed was reproductive output by counting the total number of inflorescences, which is the y-axis in this graph, because I wanted to see if the impacts of drought stress impacted reproductive output differently in plants of different ages. And I did in fact see this. There was a significant difference with the plants that were 4 weeks old at the start of the experiment to the intermediate age group, producing the greatest number of total inflorescences, indicating that the detrimental effects of drought stress had the least substantial impact in reproducing reproductive output in this age group. So to recap the key findings of this chapter, I found that drought response did differ by population and was not significantly correlated with any of the 19 bioclimatic variables I assessed. These differences may be partially explained by root surface area to above ground biomass ratio, and I also saw differences in drought stress response based on plants of different ages, where the photosynthetic rate declined most quickly in the oldest plants, and the plants of the intermediate age group produced the highest number of total inflorescences. So now I'm going to revisit the 3 shared evolutionary concepts that I introduced in my introduction and how they relate to this chapter. So the concept of variation was apparent in the diversity in morphology, drought response, and flowering I saw across these populations. Something that I saw in terms of response to context within this experiment is, though I was assessing differences between populations, I also saw differences within populations across my 2 experiments that were not the goal of the experiment, but was interesting because the first experiment was done during the summer, the second one was done during the fall. And because these plants are responsive to photoperiod, the shorter photoperiod in the fall caused the plants of the same population to flower earlier in the fall than they did in the summer, which was very interesting. In terms of anthropogenic impacts, the broader implications of this research include the importance of assessing genetic diversity in terms of stress response in wild species like Soteria viridis, which can be helpful to improve drought tolerance and stress tolerance within Soteria italica, so that agricultural species such as this one can continue to have high levels of production in spite of increasing threats from drought stress due to anthropogenic climate change. In this experiment, I overall looked at physiological responses to drought stress as measured by photosynthesis, and I was also interested in the underlying genetic and transcriptomic mechanisms that led to these physiological responses. So in my next chapter, I compared transcriptomic response of two populations of Ceteria viridis across 3 different stresses. So as I've mentioned already, evolutionary biology must consider the impacts of anthropogenic change on the biosphere and how this will impact evolutionary trajectories of different species. This includes predicted increases in drought, as well as predicted increases in soil salinity, which can occur due to a combination of sea level rise, drought, and agricultural use of land. This map shows some of the areas with the red dots in which soil salinity is already an issue, and this is expected to increase over time. Somewhat paradoxically, in the context of generally thinking of climate change as involving an overall increase in temperatures, chilling stress is also going to be an increased risk for many plant species due to the stochasticity of weather patterns that are projected under anthropogenic climate change, which can for some plants also include increased risk of chilling damage during the spring. So again, plants are unable to escape from stressful environments and instead must respond by changing their bodies in order to better withstand these forms of stress. This figure shows some of the ways in which plants perceive these different forms of stress and then mount various internal responses, including these differences in transcriptomic factors. So one of the interesting things about this figure is that it shows these multiple different forms of stress and how in some of the types of responses that plants mount, there are shared aspects of these responses in spite of the stressors being different. And so I was interested in comparing how plants would respond to different sources of environmental stress at the transcriptomic level. And again, I used Ceteria viridis, and this time I looked at 3 sources of stress: drought, chilling, and salinity stress. And I used 2 populations from my previous chapter, the China population, which had been shown to be more drought tolerant, and the Chile population, which had been shown to be more drought sensitive. And to create these stress treatments, I again used a greenhouse dry down to assess drought. To create a salinity stress environment, I watered plants with a 200 millimolar solution of sodium chloride. And to create chilling stress, I kept plants in a 4-degree cold room. I had separate sets of control plants for each of these conditions in order to create a nearly identical experience for the control plants minus the specific stress. So for plants that were experiencing intermediate levels of each of these stresses, I harvested leaf tissue and extracted RNA, and then I sequenced the full transcriptomes, and then used a bioinformatics pipeline involving FastQC, Trimmomatic, Bowtie, RSEM, EBSEq, and then I did my analyses that I'll present today using RStudio, Panther, and NCBI Blast. And so before I get into my results, if any of you have worked with transcriptomic data, if you do a full transcriptome, you have so much information that trying to decide how to analyze it is basically like walking through a forest and having to pick your 3 favorite trees. So I didn't do every possible analysis on my transcriptomic data because that would be years of work. So these are just 3 of the types of analyses I did. So something that I was interested in is differential expression of genes in response to stress. So I used a false discovery rate cutoff of 0.01 to assess which genes were differentially expressed in terms of either up or down regulation in the stress conditions as compared to the control conditions. So when I refer to differentially expressed genes, or DEGs, from here forward, that's what I'm referring to. And in these panels, the left one shows results from the Chile population, the right one shows results from the China population. And the numbers represent the number of differentially expressed genes. And note that the y-axes are different across the panels. And the blue bars are chilling stress, the red bars are drought stress, and the yellow bars are salinity stress. So getting into my results, I did see differences in the number of differentially expressed genes across populations and treatment conditions. The most striking result was the multiple levels of fold change difference in both up and down regulated genes in response to chilling stress in the China population, which is here. This was much greater than in the Chile population or any of the other responses. And I also noted differences in genes up and down regulated in response to drought stress, with the China population showing fewer up and down regulated genes and more being shown in the Chile population. Based on what we know about differences in physiological response to drought between these populations, the reduced amount of gene expression change in the China population may reflect the fact that these plants are more adapted to this stressor and less stressed overall, and therefore producing fewer changes in gene expression. And finally, with salinity stress, There were more up and down regulated genes in the China population than the Chile population, though these differences were not as pronounced. I was also interested in whether the genes that were differentially expressed across population and treatment condition were shared. And when I looked at which genes were shared in terms of regulation changes across different stress conditions, most of them were in fact not shared. So if you look at these triple Venn diagrams, the center would represent genes that were up or down regulated in more than one, or in all three stress conditions. And for genes downregulated under stress in the Chile population, none were shared between all three. For genes upregulated in the Chile population, there was one gene that was upregulated under all three stress conditions. And similarly for the China population, for both up and down regulation, there was only one gene that was consistently up or down regulated for all three stressors. So I was also interested in the function of these genes, and I did a gene ontology analysis to see which functional terms were most commonly associated with these differentially expressed genes. So what this figure is showing is the three different stressors for the China population and which of these functional terms were most commonly represented among upregulated genes. And I can only show one population at a time because this figure is so big. And even though that there are some differences in the height of the bars, the clusters in which there is the most common functional terms for each of the three stressors are shared. So for all three of them, cellular process, metabolic process, cellular anatomical entity, and catalytic activity are the most common. And now I'm going to switch to the other population, but you can see that these peaks are related to the same functional terms. So even though we are seeing lack of overlap in the specific genes that are up or down regulated across these different stressors, the functions of these genes are similar, indicating that the responses may be functionally similar, even though they are accomplished through different specific genes. I also wanted to compare my results to genes that had previously been established to play an important role in plant stress response, and I used the drought database to do this. This database includes 190 molecularly characterized plant drought stress response genes that have unique FASTA sequences, and I used this to create a local database from these drought database gene sequences and blast my differentially expressed genes across it. This is a little bit complicated, so I created a graphic for it. So for each of the populations in response to each stressor, I have a list of differentially expressed genes. I took the sequences from these genes and compared them to the database I made with the 190 sequences from the drought database gene sequences. And then it gave me this result, which is showing the number of these 190 genes that had significant alignment with these differentially expressed genes. So maximum alignment would be 190, meaning that the differentially expressed genes for each set aligned with every single drought database gene. I didn't see 190 for any of them, but I did see alignment with genes in the drought database for all of my lists, indicating that some of the functions of the genes in the drought database are also shared by the genes that I saw upregulated in response to these stressors. Another tool in the drought database is this pathway browser that shows the function of the different genes in the database, going from the most general to the most specific, and the number of genes from the drought database that have each function. So I recreated this using my differentially expressed genes, and to orient you to this figure, the number on the right shows the total number of genes in the drought database for each functional category. The number on the left shows the number of genes that were upregulated under stress in the China population that aligned with these drought database genes, and the number in the middle is the ones from the Chile population that were upregulated under stress and aligned with these genes. So I'm just going to zoom in to show you a few of them. One of them are these transporters, including the ABA importer and exporter, that genes upregulated in response to stress from both populations aligned with. ABA stands for abscisic acid, which is a very important plant hormone that's involved in stress response. And the alignment from both populations indicates that ABA is playing an important role in stress response in both populations. I also noted some differences in this analysis. So transcription factors play a very important role in plant response to stress as well. And different transcription factors have different impacts on stress response. And for some of the transcription factors in this chart, including DREB and AP2, there was alignment of more genes from upregulated under stress in the China population with these genes than from the Chile population. And for other transcription factor families, such as the MYB population, it was reversed, where there was more alignment with genes from the Chile population, indicating that these transcription factors seem to play different roles in mediating stress response across these different populations. So to recap my key findings from this chapter, I did find substantial differences in gene expression across populations and treatment conditions, as well as variability in the number of differentially expressed genes for each population and treatment condition, with the most up and down regulated genes in response to cold stress for the China population. I also saw a pattern of increased regulation changes under conditions that I believe, based on my evidence and other previous research, were the population experiencing higher stress. Even though there were differences in the specific genes up and down regulated in response to stress across population and treatment condition, it seemed that the functions these genes were serving was similar. And this was also supported by the overall alignment of upregulated genes to the drought database genes, which showed that there were some shared responses across population and treatment condition in relation to the functions of these DDB genes. And there were also differences, so indicating some shared and some differential responses at the functional level. And again, touching on these key evolutionary concepts that are running through my chapters, when we think about variation in this chapter, I saw largely unique transcriptomic responses at the genetic level across populations and treatment conditions. In terms of response to context, it was interesting to me that although the China population is from a region with cooler average annual temperatures, it showed an elevated transcriptomic response to chilling stress. However, as a short-lived annual, looking at the average annual temperature doesn't necessarily tell you the time of year and the specific temperature conditions these plants experience during the time that they're actually growing, and the specific selective pressures they face as a result. And in terms of response to anthropogenic impacts, this research is being done in the context of increasing and complex threats from multiple sources of abiotic stress, including sea level rise, agriculture, and increasing drought that will impact soil salinity, as well as the effects of drought and chilling stress. So now I'm going to go into my final chapter, which, as I mentioned, is on a somewhat different topic. It's about creating LGBTQIA+ inclusive classrooms and curricula. But again, I think there are important shared themes with my other chapters. The image on this slide is a screenshot from a documentary that I was interviewed for that came out in 2020 about the experiences of LGBTQIA+ people living in Kansas. And the screenshot is from when I was trying to answer the question. They asked me about, what did you think it would be like to move to Kansas as an LGBT person before you moved there? And so the documentary is called No Place Like Home. It's based on a book of the same name by C.J. Genovi. And this book basically discusses, given that in the American imagination, Kansas is a place that LGBT people leave, what does it mean when they stay? And something that I found living in Kansas and meeting other LGBTQIA+ people in Kansas is that there is a development of really extraordinary resilience and respond to experiences of hate and prejudice that I think is in many ways meaningfully similar to the resilience that I've seen in plants that they develop in response to just difficult environments that they are unable to leave. One example of the resilience that I saw in Kansas is that if you've ever been to the College of Arts and Sciences graduation at K-State, the Westboro Baptist Church shows up to protest it with homophobic signs, and a local human rights organization gets there earlier than Westboro Baptist Church, as early as 6 a.m. every year, in order to block these homophobic protesters from the people entering the graduation and create a positive and supportive presence. However, in spite of forms of resilience and support like this, forms of hatred and oppression continue. And just last year, the state of Kansas passed 2 anti-transgender pieces of legislation, one of which prevents trans girls from playing sports, and the other one which impacts the ability of trans people to use public bathrooms. And these forms of anti-trans hatred through legislation are unfortunately not contained to Kansas, but are occurring in many parts of the US. This map shows in blue all the states that have proposed anti-transgender legislation so far in 2024, and 24 of these pieces of legislation have passed. Something that's interesting to me is the language of these pieces of legislation. I just have 2 examples here from the bills that passed in Kansas last year, but generally this type of language is shared. And there's a lot of references to biological sex being used as a justification for transphobic oppression. And this is based on claims like biological sex referring to people being fundamentally either male or female, related to their reproductive systems and ability to produce either ova or sperm. And this is not an accurate understanding of human biological sex or gender, but I've noticed that these justifications for transphobic hate, violence, and oppression that rely on biology have been going on for years and are becoming increasingly common. And something else that I noticed during my time living in Kansas and working as a teaching assistant for biology classes is that biology courses have a unique relevance to aspects of human diversity and identity, but often the language that we use to describe certain aspects of human diversity, such as sex and gender diversity, are uncomfortably close to the same language being used to justify transphobic oppression. So there's a lot of language referring to sex as a binary that is determined by specific biological markers such as hormones, and even extending the role of these hormones to creating the basis of gender. In this case, applying this to not only humans, but also across vertebrates. And this is just an example from a commonly used biology textbook. And we also see examples like this. This is showing some different strategies for sex determination through chromosomal means. And what's interesting to me is that even though it's showing diversity in systems of sex determination, there is still this unified representation of sex as either the male, female, or male symbol or the female symbol, showing that even within diversity, this diversity is subordinate to the overall sameness of sex as either male or female across all species. And I know that this is not an accurate representation of the true diversity of biological sex across humans or across taxa. And it felt really important to me to consider whether there was a different way to teach this, given the similarities I was seeing between this language that we were using in the classroom and the language being used to oppress trans people, which personally affects me as well as my community. And so it became very important to me in order to be able to survive in Kansas and continue my scholarship as a biologist to find a different and better way to teach this. And so the rest of this chapter covers some of the things that I found with that. So I think that it's important when we teach about sex to understand that sex, in terms of sex verb, sexual reproduction, is something that can occur through many different types of recombination of DNA and contribution of genetic material from multiple gametes or multiple parents. And this doesn't actually need to involve gametes at all. We see examples of sexual reproduction in Spirogyra, a green algae that doesn't include gametes. There are also different types of gamete systems, including isogamy, in which gametes are the same size. And we see sexual reproduction with same-size gametes in algae such as Chlamydomonas. And there is also reproduction through gametes of different sizes in anisogamy, such as in mammals. And even within the category of anisogamy, which is common in plants and animals, there's still a lot of diversity in terms of how these different gametes are showing up in organisms' bodies, because evolutionarily, it's a consideration not only how are you passing genetic material to the next generation in terms of which gametes are present, but also how each individual can contribute one or more gametes to the next generation. So in animals, there is diversity in this, including gonochorism, where different individuals produce different gamete types, simultaneous hermaphroditism, in which individuals can produce multiple types of gametes at the same time, and sequential hermaphroditism, in which organisms can produce different types of gametes at different points throughout their life. There's also diversity in this when it comes to plants, including dioicy, where there are separate individuals to produce each gamete type. In monoicy, where different flowers within the same plant produce different gamete types, and plants that have flowers that are known as perfect that can produce both types of gametes within the same flower. And in addition to these differences in how sex shows up in bodies at the gametic level, within organisms that have gonochorism or dioicy, processes of sex determination vary, including chromosomal, environmental, and social-based systems of sex determination. And when we think about sex as an entire phenotype, it goes beyond just what type of gametes an organism produces and has many variations in terms of the morphologies and behaviors associated with being a certain sex or producing a certain type of gamete. I also think that it's important to have our materials reflect this. So on this side of the screen, I have this figure that I showed before, and I thought about some simple modifications that could be used to better represent this diversity in this figure. So I added some different text under panel A, and this acknowledges that in systems of XY sex determination, such as in humans, while most people have XX or XY chromosomes, some people have other karyotypes such as XXY. And I also added another panel that included C. elegans, which are a type of organism that rather than having a male and female sex determined chromosomally, the sexes are males and hermaphrodites, again, showing that there is variation and not a universal sex binary across taxa. When it comes to teaching sex in humans, there's also a frequent focus on gametes and reproduction in a way that is very related to a binary conception of chromosomes and gametes. However, when we think about how sex actually functions in terms of human culture, sex is also something that's used as a social, legal, and medical classification. And just thinking about it from a gametic perspective is not very useful because the ways that sex is actually utilized goes so far beyond this. And there are many reasons why individuals, either for their whole lives or at a certain part of their life, are not able to contribute DNA to the next generation. And they are still subjected to categorizations related to sex in terms of things like what sex categories put in an electronic health record system that determines insurance coverage for different medical procedures, and the need to have a sex marker on identification. So I think that it's important to acknowledge that human sex is a complex developmental phenotype that has a variety of different components, all of which vary along spectrums, and these spectrums interact with each other, producing a lot of diversity. And an important term for understanding this diversity is intersex variation. This refers to people who are born with physical traits that vary in ways that don't fit expectations of a biological sex binary, and people who are not intersex or endosex. And intersex isn't just one thing. It includes over 40 recognized variations that relate to variations in chromosomes, gonads, including people who have gonads that have some testicular tissue and some ovary tissue within the same gonad, as well as differences in external genitals. And intersex variation is a natural part of human diversity, but it's often been ignored or pathologized, which has contributed to the way that many intersex people experience medical harm because of the ways that intersex bodies are considered to be a social problem that needs to be fixed by medical solutions, including sometimes non-consensual medical procedures done on intersex children. So I briefly want to touch on understandings of gender within this conceptualization of an inclusive biology classroom. So I think it's important to understand the way that gender is a set of cultural organizing categories and expectations, and it varies across cultures. This is a great map showing some examples of different gender diverse cultures across the world. And notably, people in these different parts of the world and across recent history don't substantially differ in their sex characteristics, but because these conceptualizations of how many gender categories a culture has and what it means to be part of each of these categories differ, this again affirms that gender is not determined by sex traits. So finally, in addition to the content that we teach, I think the way that we teach it is also important. So being intentional about word choice and not applying human gender stereotypes to non-human animals, using specific language to talk about biological concepts, challenging inaccurate texts in the same way that we might challenge a typo in a textbook and alert students to it. We can also challenge outdated or harmful representations of human diversity. We can also offer our own diverse examples if they're not present in our texts. For example, this is a TED Talk by an intersex person about their lived experience of having androgen insensitivity syndrome, and watching this TED Talk with students could be an introduction to understanding the developmental biology of androgen insensitivity syndrome that can, for example, lead somebody to be born with both testes and a vagina. I also think it's important for instructors to consider your own pre-existing biases and understand that as much as we strive to be objective in our science, biology occurs in a political context and is politicized in ways that substantially impact certain populations. So this is just another example of how we can go from the text example I gave at the beginning that is enforcing these kind of ideas that conflate sex and gender to instead taking a different approach. So instead of saying something like there are male and female sex hormones, we could say ratios of different sex hormones affect how sex characteristics develop. I'm going to skip this example for time and just move on to my connections to these same shared evolutionary concepts. So in this chapter, we've seen how variation in sex is persistent across taxa, including in humans. In terms of response to context, thinking about the ways that terms like male and female are used, what we mean by them is really context dependent. So what it means to be a female flower is very different than what it means to be a female monkey. And in considering the anthropogenic misconceptions about biology that are used to justify laws and policies that harm subsets of human diversity, including trans and intersex individuals, and the ways that biology instructors have the opportunity to challenge these misconceptions. So now I'm going to give a brief conclusion to my dissertation research and try and synthesize it a bit. So my major findings include my exploration of interpopulation diversity and stress response in Ceteria viridis. I found differences in physiological drought response by age and populations. I also found differences in transcriptomic response to abiotic stressors across populations, including variations in functional and transcription factor regulation, and some similarities in the function of differentially expressed genes, even though the genes themselves differed. And my last chapter added to emerging discussions of LGBTQIA+ inclusive pedagogy in biology, including presenting some alternatives around how we discuss sex and gender diversity compared to standard biology texts. Some of the broader implications of my dissertation research include an emphasis on the way that current and future plant evolution will be inherently entwined with the anthropogenic climate change and environmental degradation that is predicted to progress. And this includes increases in many forms of abiotic stress that are predicted to detrimentally affect plant populations. And at the same time, plants are known to have incredible qualities to mitigate some of these effects of climate change and environmental degradation, including the ability of plants to do carbon sequestration, prevent erosion, and phytoremediation. I also hope that my dissertation research has implications for encouraging co-valuation of human diversity as well as biodiversity in the biological sciences, which includes being able to better value diverse perspectives by addressing the forms of institutional harms and inequities that have historically led to their exclusion and underrepresentation, as well as my approach to this dissertation, which has taken a meta consideration of not only doing science itself, but also the history and culture of science as a system of knowledge production, which I hope can lead to understandings of how to do more accurate and ethical science. And that brings me to my acknowledgements. And there are so many people who have helped me throughout this process, both as a scientist and as a person. I definitely don't have time to acknowledge all of them. And what's shown on screen right now is only a partial list. I really want to take a minute to thank my advisor, Mark Ungerer, for everything that he has done to support me as a scientist and a person, including being accommodating of the many challenges that I faced in graduate school and being so supportive of my decision to go in an unconventional direction with some of my dissertation work, as well as his incredible patience with me over this time. And I absolutely would not be here able to defend today without everything he's done for me. So thank you so much, Mark. I also want to acknowledge my other committee members for their support throughout this process, as well as the other faculty, staff, and grad students in the Division of Biology, my funding sources, the people who helped me with my bioinformatics and greenhouse work, my various co-authors on different publications, the amazing student and community LGBT leaders I met in Kansas, some of the amazing healthcare providers who went above and beyond to help me deal with a variety of complicated health problems while in graduate school, my colleagues and the other wonderful people I've met at FedUp, Interconnect, and in my time as an ORISE fellow, my friends and family, all of my plants. And I also want to remember my friends Stephanie Phoenix and Rexy and KT who have passed away during the time I've been in grad school. Sorry. So I have some fun pictures of some of the awesome people who have been in my life during this time. Again, this cannot represent all of them. There's been so many, and I'm grateful to everyone. And I also want to especially acknowledge my family for everything that they've done to support me, as well as my roommates, Caitlin, Chloe, and Bali, who support me every day.